Today we are going to start with the seventh topic of this series, which is megaloblastic anemia. So we know that in a cell we have a nucleus, and the nucleus has DNA. To make this DNA we need several items like vitamin B12, folate, etc. And when these vitamins are deficient, impaired DNA synthesis occurs, and impaired DNA synthesis leads to delayed nuclear maturation. So cytoplasm mature normally, but there is delayed nuclear maturation. So there is nuclear cytoplasm asynchrony. So large abnormal marrow precursors are formed which are known as megaloblasts. This leads to ineffective hematopoiesis, which will further lead to decrease in RBC production, and ultimately it causes macrocytic anemia. The RBCs are large. So this is the overall concept of the megaloblastic anemia. Now coming to the details. So first of all, what is the definition of megaloblastic anemia? We have seen that there is impairment of DNA synthesis. Why? Because there is deficiency of B12 and folic acid, which is needed for the maturation of DNA. As DNA synthesis does not occur properly, so nuclear maturation does not occur. Hence, nuclear cytoplasm ratio is disrupted and the cells will turn out to be big. This is known as megaloblastic. This will lead to ineffective erythropoiesis. So this ineffective hematopoiesis leads to anemia, and there are also distinctive morphological changes which presents as abnormally large erythroid precursors and red cells. So the two types of megaloblastic anemias occur due to B12 deficiency and folate deficiency. Both are important cofactors required for synthesis of thymidine, which is a DNA base. Folate deficiency causes folate deficiency anemia, and B12 deficiency is mostly due to pernicious anemia. So now let's look into the morphology. In the peripheral blood findings, let's first see the RBC shape and the morphology. First of all, the RBCs will be macrocytes and oval. So they are also known as macro-ovalocytes. Second of all, there will be no central pallor. So the RBCs appear hyperchromic because of increase in hemoglobin. Then there are teardrop cells which can be found, and as the sizes and the shapes are variable, therefore anisocytosis and poikilocytosis are present. Then if we look at the mean corpuscular volume, obviously it is increased as it is macrocyte. Then MHC is increased because hemoglobin is also increased, but MCHC, that is the mean corpuscular hemoglobin concentration, it is same because both size as well as hemoglobin concentration has increased. Neutrophils are also larger than the normal size, and we can find nuclear hypersegmentation. You can see in the picture there are 5 or more nuclear lobules, and also we can find nucleated RBC precursors. Now let's come to the marrow findings. Obviously it is hypercellular because of the compensation mechanism. Erythropoietin has increased due to anemia. Then there will be megaloblastic changes. For the nucleus, it retains the chromatin and also its maturation is delayed. However, cytoplasmic maturation and hemoglobin accumulation runs at the same pace. So there is nuclear cytoplasm ratio asynchrony. So DNA synthesis is hampered in almost all the cells. So there can be giant metamyelocyte band forms, which are basically the precursors of WBC, which are also seen in this kind of anemia. Also we can find nucleated megakaryocytes, which are basically precursors of platelets. Now let's learn about anemia of B12 deficiency happening due to pernicious anemia. So our bone marrow needs B12 for DNA synthesis, and our nerves also require B12 for its myelin maintenance. But B12 is a VIP. It cannot travel alone. It needs intrinsic factor, which is like a bodyguard, and it is secreted by the parietal cells of the stomach. However, in pernicious anemia, what happens? The immune system is confused. Instead of protecting the body, it attacks the gastric parietal cells or the intrinsic factors. So it's basically an autoimmune attack, and vitamin B12 cannot be absorbed without the intrinsic factor. So this results in B12 deficiency, and this further causes defective DNA and ultimately results in megaloblastic anemia. So here B12 deficiency is not a dietary deficiency, it's an absorption problem. Now let's learn about normal B12 metabolism. Our daily requirement of B12 is around 2 to 3 micrograms, and animal products have a greater source of B12 rather than plant products. So now let's look into the metabolism process. First, the food is taken and the B12 present in the food is binded to a protein. Now, pepsin is released from the stomach. This pepsin will separate the protein from the B12. Now, another element called haptocorrin, which was initially released by the salivary glands, will reach the stomach and bind to this free B12. Now, protease will come from the pancreas in the intestine. And what will happen? This will separate the haptocorrin and the B12. And this free B12 will now bind to a factor called intrinsic factor released from the parietal cells of the stomach. These two will travel together and reach the ileum. In the ileal cells, we have a receptor called the tubulin receptor to which the intrinsic factor binds, and the B12 is endocytosed. Now this B12 needs someone to carry him. So we get another protein which is called transcobalin 2. This is a major carrier protein and it delivers this vitamin B12 to liver and other cells like bone marrow, GIT, etc. Now let's look at the diagram properly once again. So when the food is taken, B12 is attached to a binding protein. When it reaches the stomach, pepsin is released and it separates the protein from B12. Haptocorrin, which was released from the salivary glands, reaches the stomach and it binds to the free B12. They travel together to the intestine where pancreas secrete an enzyme called protease. This protease separates the haptocorrin and the B12. Now this free B12 will at last bind to intrinsic factor which was released by the parietal cells of the stomach, and they will move together to the ileum and attach to the tubulin receptor. Here the B12 is endocytosed and then it is further attached to a major carrier protein known as transcobalamin 2. This transcobalamin helps to deliver the vitamin B12 to other places. Now what are the biochemical functions of vitamin B12? Inside the cell, methyl tetrahydrofolic acid uses methionine synthase and converts into tetrahydrofolic acid. In this process, methyl from methyl tetrafolic acids gets attached to cobalamin, that is B12, and gives rise to methyl cobalamin. Now simultaneously there is another reaction where homocysteine uses methionine synthase to become methionine. Now this reaction needs a methyl group and it derives the methyl group from the methyl cobalamin which gets converted to cobalamin again. Now let's see why this reaction is so important to us. This tetrahydrofolic acid uses an enzyme called thymidylate synthase and converts into methylene tetrahydrofolate. This methylene tetrahydrofolate is very important to convert deoxyuridine monophosphate to deoxythymidine monophosphate. This is required for DNA. It's a base that is used in the DNA. The conversion of homocysteine to methionine is very important as it regenerates B12. B12, also known as cobalamin, is converted to methyl cobalamin when methyl tetrahydrofolic acid converts to tetrahydrofolic acid. Therefore, if there is no B12, then tetrahydrofolic acid levels will decrease and the levels of methyl tetrahydrofolic acid will increase. Therefore, folate will be trapped in the form of methyl tetrahydrofolate. This is very important for the exams. If sufficient amount of tetrahydrofolic acid is not present, then deoxythymidine monophosphate will not be formed, which is a very important base for the DNA, and megaloblastic changes will start to happen. Now let's summarize. 5-methyl tetrahydrofolate gives the methyl group which is carried by vitamin B12 and it is given to homocysteine so that it can become methionine. In this process, tetrahydrofolate is also regenerated and it helps to make the DNA base pair, that is thymidine, and then DNA synthesis is normal if all this process happens in a fruitful way. Now, if B12 is deficient, then methionine synthase enzyme will not work because B12 is the cofactor. Therefore, tetrahydrofolate cannot regenerate and functional folate deficiency develops because folate is trapped as 5-methyl tetrahydrofolate. Okay, so now as THF or tetrahydrofolate is not there for thymidine formation, then DNA synthesis decreases. So this is the overall story. Now let's learn about the second function, which is about methyl malonyl CoA reaction. In these reactions, nerves are involved, so the manifestations are neurological. So in this reaction, methyl malonyl CoA converts into succinyl CoA using the enzyme methyl malonyl CoA mutase, and the cofactor here is B12. This succinyl CoA is used in fatty acid metabolism and myelin stability. Now, if B12 is deficient, then methyl malonic acid will accumulate and abnormal fatty acids will form because succinyl CoA is deficient. Therefore, myelin damage occurs and we get neurological symptoms. Now this myelin sheath degenerates in the posterior columns, lateral corticospinal tracts, and hence give rise to numbness, tingling, gait, and loss in vibration senses. Now why folate deficiency does not cause neurological symptoms? This is because folate is used in the metabolism of DNA synthesis, as we have already seen, but it is not used in the metabolism of methyl malonate CoA. Therefore, folate deficiency causes only anemia. However, B12 deficiency causes anemia as well as neurological diseases. Now let's learn about the pathogenesis of pernicious anemia. So what is the definition? It is a specific form of megaloblastic anemia. And how it is caused? It is caused by autoimmune gastritis. This impairs the production of intrinsic factor, which is very much required for the B12 uptake from the gut. So autoimmune reaction develops and the immune system attacks the gastric mucosa, and prolonged attack will lead to chronic atrophic gastritis. Okay, this will destroy the parietal cells, and parietal cells nahi hoga to intrinsic factor bhi nahi hoga, and so intrinsic factor cannot bind to vitamin B12. Therefore, B12 absorption in the terminal ileum will not happen, and this results in B12 deficiency. Now, this autoantibodies can be of three types. Type 1, which is most commonly found, stops the binding of B12 to the intrinsic factor. And type 2 autoantibodies stop the binding of the B12 intrinsic factor complex to the tubulin receptors. Therefore, here intestinal absorption is being affected. And the type 3 autoantibodies recognize the alpha and the beta subunits of the gastric proton pump, which are present in the parietal cells. As autoimmune diseases can also affect the other organs, therefore pernicious anemia is associated with other autoimmune disorders like autoimmune thyroiditis and autoimmune adrenalitis. Now, what are the other causes of B12 deficiency? If we look at the entire metabolism pathway of B12, we can see that various steps can go wrong. For example, if there is decrease in the HCl production, then pepsin secretion will also decrease, and therefore separation of B12 from the binded protein will not happen. Also, there can be insufficiency of exocrine pancreas, so B12 cannot be released from the haptocorin B12 complex. Then if there is gastrectomy, then intrinsic factor can be lost. Also, if there is ileal resection or diffuse ileal disease, it prevents adequate absorption of the complex. Other factors like tapeworms or raw fish compete with the host for B12. This also causes deficiency. Factors like pregnancy, disseminated cancer, hyperthyroidism, all this produce relative deficiency even when absorption is normal. Now let's look at the morphology. The peripheral blood findings and the bone marrow findings are same like the megaloblastic features that we have discussed earlier. Okay, now let's look at the other alterations. There will be fundic gas atrophy. So there will be damage to the chief cells and the parietal cells. The glandular epithelium will be converted to mucus secreting goblet cells like those lining the large intestine. This is known as intestinalization. And the tongue will become shiny, glazed and beefy because of atrophic glossitis. So in these conditions, parenteral B12 administration would not help because it's an autoimmune problem. And now what happens in the CNS? In the CNS, demyelination occurs in the dorsal and the lateral spinal tracts. So there will be loss of axons. So this will lead to symptoms like spastic paraparesis, sensory ataxia and severe paresthesia in the lower limb. So what's the summary of the clinical features? We can find hematological features, gastrointestinal features and neurological features in pernicious anemia. Now coming to the lab findings, first of all there will be moderate to severe megaloblastic anemia due to increase in the mean corpuscular volume. Secondly, there will be leukopenia and we will find hypersegmented granulocytes due to defective DNA synthesis. As absorption will be defective, therefore serum B12 levels will decrease. However, serum homocysteine and methylmalonic acid will increase. Okay, serum antibodies specific to the intrinsic factor can also be found. Now, what's the treatment? Basically, the treatment is parenteral B12 administration, and after 5 days, we have to check if the reticulocyte count and the hematocrit levels have increased or not. So this B12 administration will be reversing the anemia and the neurological diseases. However, the gastric mucosa changes and the risk of carcinoma remains unaffected because it is caused by the autoimmune system. So that was all about B12 deficiency. Now coming to anemia due to folate deficiency. So what is folate? Folate is basically vitamin B9. This folate is converted to dihydrofolate, which is further converted to tetrahydrofolate using the enzyme dihydrofolate reductase. This tetrahydrofolate is active and it carries 1 carbon groups which is used in the synthesis of purine and thymidine. So it is basically useful for DNA synthesis and it results in normal cell division. Therefore, if there is deficiency of folate, then tetrahydrofolate will not be formed and therefore purine and thymidine synthesis will be deficient. So DNA synthesis will be impaired and normal cell divisions won't happen, resulting in megaloblastic anemia or macrocytic anemia. Now let's look at the etiology. So it can happen due to 3 reasons, either due to decreased intake or due to increased requirement or due to impaired utilization. Decreased intake can happen due to deficient nutrition, then impaired absorption and alcoholism. Generally, these are accompanied by the general malnutrition symptoms like cheilosis, glossitis, dermatitis, avitaminosis, etc. Features of malabsorption like sprue, then drugs like phenytoin, oral contraceptives can also contribute to it. Increased requirement can be due to pregnancy or in infants or due to hyperactive hematopoiesis like in the case of chronic hemolytic anemia or in disseminated cancer. However, impaired utilization can happen due to intestinal diseases that restricts the absorption of folate. Now coming to the mechanism of an important drug which is methotrexate. This methotrexate is a folic acid antagonist. It inhibits dihydrofolic acid reductase. Therefore, it reduces the level of tetrahydrofolate which was very important for DNA synthesis and hence it affects all rapidly proliferating tissues like bone marrow and GIT. Now let's compare the clinical features of B12 and folic acid deficiency. Megaloblastic anemia is present in both of them. However, neurological symptoms are present only in B12 deficiency. Methylmalonic acid level is increased in B12 deficiency, however it is normal in case of folic acid deficiency, and homocysteine levels are increased in both B12 as well as folic acid deficiency. Therefore, the features are macrocytic anemia, hypersegmented neutrophil as we have learned earlier, then low serum folate and high serum level of homocysteine, and methylmalonate levels will be normal. So that was all about megaloblastic anemia. Now I will want all of you to go and read the same thing from Robbins.